Today, we're going to dive into a really remarkable story of scientific discovery. Seriously, it's about a mysterious disease, a microscopic battle that's happening inside the human body, and this brilliant quest to re-engineer one of our own proteins to fight back and actually save lives. I mean, just think about that for a second. The very system that's designed to protect you, your own personal army against germs and viruses, suddenly gets confused and starts a campaign of friendly fire against its own leadership. Now, this isn't science fiction. For some people, it's a terrifying reality, and it's the very start of our story today. So here's how we're going to follow this journey. We'll start with the betrayal by the body, then meet the crippled general of our immune system. After that, we'll see how scientists cracked the code of the attack, designed a clever stealth solution, and finally, we'll look at the race to get this whole thing from the lab to a patient's lifeline. Okay, so our story really kicks off with a medical puzzle. Doctors started seeing these adult patients who were seemingly out of nowhere getting hammered by infections that a healthy immune system would just laugh off. It was some new kind of immunodeficiency, but nobody knew what was causing it. It eventually got a name: Adult-Onset Immunodeficiency, or AOID. And what makes this thing so weird is the cause. We're not talking about a virus or a genetic defect someone was born with. Nope. The patient's own body was manufacturing these rogue antibodies, they're called autoantibodies, that were specifically targeting and shutting down a critical part of their own defense system. And the stakes here, they could not be higher. For patients with AOID, the mortality rate is a staggering 25%. That's a 1 in 4 chance of not making it. This wasn't just some scientific curiosity. Finding an answer was literally a matter of life and death. You see, without their defenses, patients were just left wide open to this barrage of what are called opportunistic infections. These are germs that are usually pretty harmless, right? But they see an opportunity, a weakened immune system, and they pounce, causing all sorts of devastating illnesses, from really severe bacterial infections to dangerous fungal diseases. So, the big question was, what exactly were these rogue autoantibodies attacking? To solve the mystery, scientists had to figure out the specific target, and it turned out to be a protein that acts like a 5-star general for our entire immune system. And that general has a name: Interferon gamma, or IFN gamma for short. Think of it this way. Your body has these soldier cells called macrophages, and they are fantastic at gobbling up and destroying invaders. But they don't just act on their own. They need orders. Interferon gamma is the protein that shows up and gives those orders: activate, seek, and destroy. And this slide right here shows the core problem perfectly. On the left, in a healthy person, the general IFN gamma gives the command, and the macrophage soldiers get right to work. Simple. But on the right, in an AOID patient, you've got these autoantibodies acting like a form of friendly fire. They intercept and neutralize the general before the message can ever get through. So the soldiers, they're just left standing around, inactive, while the enemy completely overruns the place.
All right, so the scientists knew what was being attacked. But if you're going to fight back, you need to know how. This kicked off an incredible piece of scientific detective work to figure out the enemy's exact strategy.
This was the million-dollar question. You see, a protein is this long, complex, folded-up chain. The autoantibodies weren't attacking the whole thing. They were zeroing in on a very specific spot, a kind of weak point. Finding that exact location, it's called an epitope, was the absolute key to everything that would happen next. And to find it, they brought out this really impressive scientific toolkit. I mean, they used techniques that let them test millions of tiny protein fragments to see which ones the antibodies would stick to. They compared genetic codes to find patterns. They even started engineering their own mutant versions of the protein just to see what changes might block the attack. And you know what? It worked. After all this painstaking research, they had their aha moment. They had created a precise map, pinpointing the exact amino acids on the interferon gamma protein that the rogue autoantibodies were latching onto. They had cracked the enemy's code. And this is where the whole story really pivots, because knowing the enemy's strategy allowed their researchers to design this brilliant, two-pronged counterattack. This is where basic discovery turns into a life-saving innovation. First up, they tackled diagnoses. The old way of testing for these autoantibodies was this complex lab test that was slow and it was expensive. So, using what they now knew, they developed a rapid strip test, kind of like a home pregnancy test. This meant doctors could get answers way faster for more people, which is absolutely critical when you're dealing with a fast-moving disease. And second, and this is the real game changer, they created a new therapy. On the left, you've got the original protein, easily spotted and neutralized. But on the right, that's their creation, a mutant, re-engineered interferon gamma. By just slightly changing the exact spot where the autoantibodies bind, they created a stealth version. It's like a general in disguise. The friendly fire can't even recognize it, but, and this is so crucial, it can still deliver its orders to the soldier cells. These innovations were so new, so important that they led to two separate patents, one for that diagnostic test and one for the modified stealth protein itself. I mean, that's the official stamp of a true breakthrough right there. But look, a discovery in the lab, even a patented one, isn't a medicine you can get at a hospital. The final chapter of this story is all about navigating that long and complicated road from a laboratory breakthrough to a real-world lifeline for patients. This whole process is often measured in what are called technology readiness levels, or TRLs. The journey starts with basic research. Then you have to prove the concept works. Then you move to developing and testing prototypes. And finally, you get it ready for commercial production and clinical trials. Every single one of those steps is a massive undertaking. And believe me, that path from the lab to the clinic has some major hurdles. For instance, when you try to produce complex proteins in huge batches, sometimes they misfold and clump together, becoming insoluble and totally useless. A key challenge for the scientists right now is perfecting that manufacturing process to get as much of the correctly folded, soluble protein as possible, the stuff that can actually be used as a medicine. So all of this work, the detective work, the engineering, the manufacturing, it's all driving toward one single vital goal: to scale up production and get this life-saving molecule approved as an official medicine that can finally be given to the patients who so desperately need it. When you step back, this whole endeavor is just a testament to the power of scientific persistence. It takes us from a baffling, deadly disease to a super-specific, intelligently designed therapy. It's a perfect example of how understanding biology at the deepest level allows us to go in and fix it when it goes wrong. And that leaves us with a really fascinating question to think about. We just saw how scientists could subtly edit one of our body's own command proteins to beat a disease. As our ability to understand and manipulate these molecules gets better and better, could this kind of biological reengineering become a whole new pillar of medicine for all sorts of conditions? It's a pretty exciting glimpse into the future.